Variant 2, June 23, paper 4, variant 2. The diagram is a cross-section of a leaf. Let's remember the layers. This is the cuticle. This is the upper epidermis. This is the palisade mesophyll, spongy mesophyll, lower epidermis. And of course, these are the guard cells, and this is the stomata. What was the question? Why the leaf is considered to be an organ? I will tell him the definition of the organ because it is made up of different tissues. This is one tissue, this is the second tissue, third tissue, fourth tissue. It is made up of different tissues that works together to do a specific function. Different tissues working together to do a specific function. Okay. Explain how layer B and C are adapted. Layer B, the first mark is to tell him what are layer B and C. What are their names? Layer B is the palisade mesophyll. Layer C is the spongy mesophyll. How are they adapted? Number one, the palisade is vertically arranged on the top side of the leaf. Why is this? To absorb as much sunlight as possible. They also have many chloroplasts to carry out the photosynthesis process. I can also say that the vacuole is expanded. What is the benefit? Vacuole expanded, so it pushes the chloroplasts to the edges to be able to carry out the photosynthesis faster, absorb more light. Therefore, palisade is B is the palisade mesophyll, vertically packed immediately below the upper epidermis, absorb as much light as possible. Many chloroplasts. El vacuole pushes the chloroplasts to the sides. Now, what about C? C is the spongy mesophyll. It has many air spaces to allow gas exchange. Also, the air spaces helps the evaporation of water from the cells to the air spaces. Type. This is enough. However, you can add these. You can add the adaptations in the xylem and the phloem because xylem and phloem here, they are available in the spongy layer. So I can tell him the adaptations of the xylem or the phloem. يعني xylem has lignin مثلا for support. It is hollow from the inside to allow the passage of water. But the above points are already enough. A student placed a plant in very hot room for 12 hours. There was bright light and it was not given any water. Hot room, bright light, no water. What will happen? The plant will lose a lot of water and it will wilt. The figure shows a series of sketches of the stomata. This is how the stomata looked like, and this is how it looked like after 12 hours. Identify the cell labeled D. The stomata is in the middle. What is D? It is the guard cell. What is the main function of stomata? It allows the gas exchange, carbon dioxide and oxygen, also needed for transpiration. Either gas exchange or transpiration. What is the advantage of to the plant of the change shown? What is the advantage of closing the stomata in the hot weather or in bright light? When you close the stomata, there will be no loss of water. You will decrease the loss of water, so you will prevent wilting. بصوا بقى, this was a tricky question. The student increased the humidity in the room and repeated the investigation. We agreed that when humidity increases, transpiration decreases. They are opposite. Predict and explain the effect of high humidity on the stomata. Take care. He is not asking you the effect on the plant. He is asking you the effect on the stomata. Effect on the stomata. That's a new question. When there is high humidity, Okay? There is less water vapor concentration gradient. The difference between the concentration of water vapor in the leaf and outside is small. So the guard cells do not lose water. Guard cells of the stomata does not lose water as before. And therefore, they will not become flaccid. They will stay turgid. Or it takes longer to become flaccid. Question 2, diagram of part of the DNA molecule. Draw a circle to identify one pair of bases. In nitrogen bases is what is in the middle, what's holding the two strands together. Any pair, you can circle any pair of bases, okay? The percentage of T in the DNA of a species is 29. طيب, this question usually comes in paper 2. Here he got it in paper 4. Let's see. Percentage of the T is 29. What is the percentage of G? How am I going to do this? We know that T binds to A. So if T is 29%, it should be equal to A, which is 29% too. يبقى التوتال of A and T is 58%. This is the total for A and T. طب what about G and C? G and C binds together. How much are they? You will get the remainder. يعني I will subtract 100 from 58. It will be 42. 42% for both of them. They are both equal to each other. يبقى each of them will be 21%. This is 21% and this is 21%. State the name given to a length of DNA that codes for a protein. Length of DNA that codes for a protein is the gene. How the proteins are made, similar to the question in variant 1, how the proteins are made. I will describe the steps which we took in chapter 18, in 17, sorry. mRNA takes a copy of the DNA. The DNA stays in the nucleus. mRNA moves to the cytoplasm and then it goes out to the ribosomes. Ribosomes arranges the amino acids according to the sequence of bases in the DNA. Okay. DNA controls the cell function by controlling the production of proteins. State 2 proteins found in the cell membrane. What are the proteins found in the cell membranes? We took carrier proteins in the cell membrane that is used for the active transport. I can also say receptor proteins, the receptors of the cells for the neurotransmitters, for example, are found on the cell membranes and they are made of protein. You can also say enzymes. Sometimes the enzymes are in the cell membranes. Question 3. Antibiotic resistance is an increasing problem worldwide. Erythromycin is an antibiotic. Figure 3.1 shows the daily doses of erythromycin per 1,000 people and the number of bacterial infections resistant to erythromycin per 1,000 people. Daily doses and the number of bacterial infections resistant. What can I see? Calculate the percentage change in the number of bacterial infections between 1993 and 1995. Where is the number of bacterial infections? They are the light gray bar. 1993 and 1995. Take care. You have to look at this scale. This is the number of bacterial infections. Here, it is the doses of erythromycin. So when I read the number, I have to read from the scale on the right. This is 180 and this is 100. How did it change? Final minus original over original times 100. I will subtract 100 minus 180 over 180 times 100. It will give me 44.4. He wants it to the nearest two significant figures, so I will write it as negative 44. Negative because it decreased. It was 180 and then it changed to 100. Okay, describe the data. How do I describe the numbers? You have two sets of data. You have the doses of the erythromycin and you have the bacterial infections. What can I say? The doses of erythromycin peaks in 1989, while the peak in the infection was in 1993. This is the peak for the doses of antibiotic, and this is the peak for the resistant bacteria. Now, this makes sense. Usually, the effect of increased antibiotic will not be shown in the same year. It will be shown after a few years. And this is what happened. When the antibiotics increased a lot, after 4 years, the resistance increased a lot. The effect appears after a while. So we say that there is a delay. There is a delay in effect. يبقى the peak of infections in 1993, which was 180 bacterial infections per 100 people. There is a delay of 4 years. No cases of resistant infections until 1991. Until here, there was no resistance. Resistance started in 1991. This is when the mutation happened. Suggest the reason for the change in number of bacterial infections resistant from 1993 to 1995. Why did the resistant infection decrease here? It's a suggest question. So you can say maybe they stopped using a lot of antibiotics, or not maybe, what they actually did. There is the erythromycin doses, it decreased. More awareness about the use of antibiotics. Development of new antibiotics, so they started to kill the resistant bacteria. Anything that makes sense. This question, explain how the bacteria becomes resistant to antibiotics. How bacteria became resistant to antibiotics? How did they develop the resistance? By natural selection, mutation occurred, which causes variation in the bacteria. The non-resistant bacteria will die when you take antibiotics. However, the resistant will survive, reproduce because it has an advantage, and it will pass the alleles to the offspring. Okay? Bacteria are prokaryotes. I want features of all prokaryotes. He did not say distinguishing features. He just said features of all prokaryotes. So what can I say? Anything present in all bacteria. I can say cytoplasm, I can say cell membrane, I can say cell wall, I can say circular DNA, anything that is present in all bacteria. Some bacteria have flagellum. What is the function of the flagellum? It allows the bacteria to swim out for movement. Okay, swimming or movement. Question 4. A student recorded the information about an aquatic habitat. Aquatic means underwater. Aquatic plants provide food for freshwater shrimps and midges. Mayflies also feeds on aquatic plants. He gave me the food web and he wants me to draw it. Construct a food web. Let's start with the first sentence. Aquatic plants provides food for shrimps and midges. يبقى I put the aquatic plants at the bottom and I drew two arrows. Food for shrimps and midges. Mayflies also feed on aquatic plant. I drew a third arrow and I wrote the mayflies. It does not have to be in this sequence. Yani if you put the mayflies here, it's still correct. And then the mayflies provide food for salmon. I drew an arrow. Oh, salmon eats the mayflies. Stoneflies feeds on the midges. and are eaten by salmon. Salmon also eats the freshwater shrimps. This is the stoneflies. They feed on midges and they are eaten by salmon. Salmon also eats the shrimps. So I drew an arrow from the shrimps to the salmon. Herons feeds on salmons also, so I drew an arrow from the salmons to the herons. And this is my food web. Do not draw the organisms. I just want the names. Complete the table using the information, identifying the names of the missing trophic levels and the organism. Primary consumer, of course, it is one of these, the first ones after the aquatic plants, shrimp or mayflies or midges. Secondary consumer can be stonefly or can be salmon. Tertiary consumer, I completed by the names. Tertiary, which is the third consumer. Quaternary, which is the fourth consumer. Tertiary consumer is it can be the salmon if I follow this food chain, midges, stoneflies, then salmon. So here it is the quaternary consumer, or it can be the herons. Aquatic plant, shrimps, salmon, herons. So it can be herons, quaternary consumer. Tertiary consumer, sorry. Tertiary from here. Aquatic plants, mayflies, salmon, herons. This is a tertiary consumer. If I follow this food chain. Herons, if I followed the food chain in the middle, this will be 1, this will be 2, 3. Yani this is a primary consumer, secondary consumer, third consumer. Okay? Primary consumer, secondary consumer, third consumer. But if I followed this food chain, it is still a tertiary consumer. Here is the producer, primary consumer, secondary consumer, third consumer. طب if I follow this food chain بقى, plants is the producer, midges is the primary consumer, stoneflies is the secondary consumer, salmon is the tertiary consumer, herons is the quaternary consumer. يبقى when he asks what organism eats in two trophic levels, it can be the herons, or which organism is the quaternary consumer, it will be the herons. Outline how the energy in the primary consumer in this aquatic food web is used to produce biomass in the secondary consumer. The energy in the first consumer. How does it go to the secondary consumer and how does it form mass in the secondary consumer? When the secondary consumer eats the primary, he takes the energy from it. What form of energy? Chemical energy. Food is chemical energy. The food is digested into small soluble molecules such as glucose, which is used in respiration for releasing energy and for making proteins, growth on making new cells. This is the new mass formed. Primary consumer contains chemical energy. Energy is transferred to the secondary consumer by ingestion. The food that you eat when you're a secondary consumer is digested to provide glucose for respiration, release of energy, and then the energy is released for growth and to make new cells. Humans also eat salmon. Predict the impact on feeding relationships of over-harvesting salmons. If we over-harvest salmons, what is the impact on this food web? First of all, the herons will decrease because they will not find food to eat. The shrimps and the mayflies and the midges and the stoneflies, the shrimps and the mayflies and the stoneflies will increase because nothing is eating them. When the stoneflies increase, the midges will decrease because it is eaten. Okay? يبقى this is the 3 points and I can write. Herons decrease because they have less food, more mayflies and stoneflies, less salmon to eat them, fewer midges as there are more stoneflies eating them. Describe what is meant by decomposer. Decomposer is the organism that gets its energy from dead or waste organic material. Animals such as salmon can be farmed for meat. Explain why it is more energy efficient for humans to eat plants than to eat the livestock. Why is it better for us to eat plants than eat meat? This is where you will talk about the energy loss. As we go along, طبعا plants are at a lower trophic level than the livestock. Yani plants are producers, while the animals may be primary or secondary consumers. So, as you go along the food chain, energy is lost between trophic levels. 90% is lost, only 10% is transferred. It is lost as heat energy in respiration, in excretion. Also, not all materials are eaten and digested. If I give him examples about how energy is lost. Okay, that's a repeated question. Question 5. The photomicrograph of a structure found in the animal and the plant cells. We took the structure before. The oval structure with foldings inside. This is a mitochondria. This is the mitochondria. What is its function? Aerobic respiration, release of energy. State the two pieces of information needed to calculate the actual length of the structure. Actual length. I need the image and the magnification. Image length and magnification to calculate the actual. The actual length is 0.00075. Converted to micrometers, I will multiply by 1000, multiply by 1000 from millimeter to micrometer, so it will be 0.75 multiply by 1000. Question 6. A student investigated the plant growth responses in the roots and the shoots. They used this method. Damp cotton wool was placed in 2 petri dishes. 3 bean seedlings were attached to the cotton wool in each petri dish. 3 bean seedlings attached. Each seedling was oriented so that the roots pointed in a different direction. Dish 1 was kept in a fixed position. The clinostat is not rotating. The motor is not rotating. It is fixed. While dish 2 continuously rotated. Both are kept in the dark. Both are kept in the dark. Okay? After 2 days, the seedlings were observed. This is dish 1. This is dish 2. Okay. What happened? What happened? Describe the pattern of growth of the roots and the shoots in 1. Describe the growth. What happened? All the roots grew downwards. Oh, the root grows downwards, root grows downwards, root grows downwards. The stem, the shoot grows upwards, upwards, upwards. No matter how you put the seed, all the roots grow downwards, all the shoots grow upwards. Okay? You have to complete the answer. It has to be because he asked about the roots and the shoots, and all the shoots grew upwards, which is negative gravitropism. What is the response? The response of the roots and the shoots here is to the gravity, gravitropism. Tab, why can't I say phototropism? Because he told you that he put it in a dark room. He kept it in the dark. There is no light. So this is not a response to light. It is a response to gravity. Response to gravity. Okay? Explain how. This is the explanation question back. How the auxins causes the difference in the pattern of growth shown by the shoots in seedlings B and E. Shoots. Here he's talking about the stem. Why did the stem here always go upwards? while the stem in the other dish grew horizontally in the same direction as it was. It did not go upwards. Why is this? Type. What controls the growth in the directions is the auxins. So I will start by telling him that auxins are formed at the tip of the shoot and then they diffuse downwards. Where do they accumulate? They accumulate on the lower side, which is closer to gravity, not on the shaded side. I will not say shaded here because there's no light. These seedlings were in the dark, so they diffuse downwards, stimulate the elongation of cells. In B, they accumulate on the lower surface of the shoot, stimulating elongation on the lower side, so the shoot bends upwards. So what about E? E was rotating. So the gravity was the gravity was equal from all the sides. Therefore, I have equal distribution of auxins, and therefore the root grows horizontally. Okay? Seeds require oxygen and water to germinate. State one other environmental condition that affects germination. Water, oxygen, he already gave you. So the third one is the suitable temperature. Suggest why oxygen and water are required for germination. You need oxygen for aerobic respiration, and water is used as a solvent for all the organisms. Water is used as a solvent, okay, to activate the enzymes, dissolve the enzymes inside. Okay, the last question was a complete question. Easy question. Enzymes are involved in chemical digestion, produce small soluble molecules that can be absorbed. Enzymes break down the molecules from large insoluble to small soluble. Two examples of protease enzymes are pepsin and trypsin. Pepsin, produced in the stomach, requires acidic conditions by the release of hydrochloric acid. Also kills harmful pathogens. This is the function of the hydrochloric acid, optimum pH, and kills pathogens. The small intestine produces trypsin, which breaks down proteins in alkaline pH. This condition is created by a substance called bile. Why did I choose bile here? Because it said it neutralizes the gastric juice, also has an important role in the emulsification of fats and oils. Yani I cannot say sodium hydrogen carbonate, although it gives an alkaline pH, but the sodium hydrogen carbonate is not used in emulsification. So here I said bile neutralizes the juice and causes emulsification of fats.